تعالوا نكمل السكند فيديو. يبقى احنا كده خلصنا السايكليك AMP اللي هو اول سكند ماسنجر كونكتد لي او باوندد للريسبتورز الجي بروتين كابلد ريسبتور. تاني سكند ماسنجر بيشتغل او coupled with الجي بروتين coupled receptor هو السايكليك GMP او السايكليك جوانوزين مونوفوسفات. اتفقنا على الابريفيشنز ان احنا هنقولها مرة ونكمل بالابريفيشن. This is unlike الcyclic AMP that it's concerned with hormonal regulation of two peptides or two hormones, الاتريال natriuretic peptide والنيتريك oxide. يبقى هو it's limited signaling pathway include hormonal regulation of atrial natriuretic peptide receptors in which الجوانايل cyclase بيبقى transmembrane guanyl cyclase, and also concerned with activation of the soluble form of guanyl cyclase by nitric oxide. يبقى هو once atrial natriuretic peptide connected to its specific receptors it will activate the transmembrane guanyl cyclase to produce cyclic GMP. Once a nitric oxide activate its receptors it will activate the soluble form of the guanyl cyclase to produce the cyclic GMP. يبقى هو unlike cyclic AMP, cyclic GMP only concerned with few cell types of a signaling pathway that regulate nitric oxide and atrial natriuretic peptide. Some effects of cyclic GMP will be mediated by protein kinase اسمها ايه بقى؟ PKG اللي هي cyclic GMP protein kinases. يبقى once cyclic cyclic GMP produced, it will activate certain protein kinases called protein kinase G or BKG to phosphorylate the desired target proteins to produce the desired biological response. تعالوا برضو نشوف الفيجر. ده هنا فيجر فور الاتريال natriuretic peptide، وهنجيب فيجر للنيتريك اوكسيد. قلنا ان الاتريال natriuretic peptide act on الجوانايل cyclase اللي هو الترانسممبرين جوانايل cyclase، okay, which convert GTP into cyclic GMP, which is the second messenger. The cyclic GMP will activate protein kinase G that phosphorylate the target protein. وهنا in this case the atrial natriuretic peptide will act on lipid oxidation, stimulate lipid oxidation, energy expenditure, mitochondrial uncoupling, and also will decrease lipolysis and release of fatty acids from adipocytes, and it aims to decrease blood pressure and sodium reabsorption. So it aims to decrease blood pressure. It's diuretic, natriuretic, and decrease blood pressure. This is the desired action of natriuretic peptide. The second figure هنا برضو natriuretic peptide atrial natriuretic peptide connect to its certain receptor اللي هي ال G protein coupled receptors. ال G protein coupled receptors will activate ال guanyl cyclase اللي هو transmembrane guanyl cyclase. تمام. will convert ال GTP into cyclic GMP. Cyclic GMP will activate certain kinases منها البروتين kinase G, okay, to produce its target action and also activate target genes. Once الأكشن ended, الcyclic GMP will be hydrolyzed by phosphodiesterase اللي هو the same enzyme like cyclic AMP and converted into 5-GMP. Okay, this is termination of the action. The natriuretic peptide will affect the skeletal muscle. It increase الميتوكوندريال content والميتوكوندريال lipid oxidation قلناها في الفيجر, and it aim to decrease blood pressure by its action like natriuretic, decrease sodium reabsorption, diuretic, and vasodilatation. This will maintain or control the blood pressure and fluid hemostasis. So the major physiological effect of the atrial natriuretic peptide is to decrease the blood pressure. The second hormone which acts on the cyclic GMP is the nitric oxide. Nitric oxide is gas. It's diffusible from adjacent nearby cells. Nitric oxide له many actions. It's bactericidal, okay, against viral and bacterial infections, يعني protective للسل, لكن هو very toxic. ما ينفعش يفضل في السيلز, علشان كده it has to be detoxicated. النيتريك oxide بيجي from the adjacent cell and diffuse inside the cell to act on the soluble form of guanyl cyclase enzyme, which convert GTP into cyclic GMP. And cyclic GMP زي ما لسه قايلين it will activate certain protein kinases اللي هي specific PKG and will phosphorylate also the desired proteins to produce its action اللي هي mainly muscle relaxation and blood vasodilatation. Okay, it will dilate the blood vessels. When the action ended, it will be hydrolyzed by phosphodiesterase into 5-GMP to terminate the signaling pathway. Another figure for nitric oxide. هنا from adjacent cells, لو السيلز فيها infection, bacterial infection, it will release from adjacent cell to enter the cell to the soluble form of guanyl cyclase. It will produce conformational changes in the enzyme, converted GTP into cyclic GMP. Cyclic GMP will stimulate certain kinases اللي هي protein kinase G BKG and certain proteins to produce its desirable action اللي هي relaxation of smooth muscle, vasodilation and inhibition of the platelet aggregation. It will be hydrolyzed by phosphodiesterase to terminate the action of nitric oxide. The third one acting on the G protein coupled receptor is calcium and phosphatidyl inositol or both يعني calcium or phosphatidyl inositol or both can act in the same signaling pathway as secondary messenger. الانزيم هنا اسمه فوسفولايبيز سي. يبقى هنا دي الليجند تمام اللي هي الهرمون will connect to الجي بروتين coupled receptors will stimulate الإفكتور اللي هو الفوسفولايبيز سي. فوسفولايبيز سي enzyme will act on the substrate. اسمه ايه السبستريت is phosphatidyl inositol diphosphate or biphosphate تمام will convert it into diacylglycerol and an inositol triphosphate. هنا الأسامي شوية طويلة هنقول تاني الأسماء بعد كده هتلاقوها abbreviations. okay يبقى تاني once a ligand a specific ligand or hormone bind to a specific G protein coupled receptor will activate the G protein will activate the effector اللي هو الفوسفولايبيز C enzyme. This will convert الفوسفوإينوسيتول biphosphate اللي موجود in the inner side of the plasma membrane into diacylglycerol and phosphatidyl and inositol triphosphate, and both act as second messenger. okay diacylglycerol unlike inositol triphosphate is highly lipophilic, so it's membrane bound. okay and it will bind to specific protein kinases or activate certain protein kinases called BKA, BKC, which will migrate to the cytoplasm and phosphorylate the target proteins to become activated. These kinases have role in signal transduction and different cell function. ده بالنسبة للدايأسايلجليسرول. يبقى الدايأسايلجليسرول لأنه highly lipophilic will stay within the membrane and will activate certain protein kinases called BKC, which will migrate to the cytoplasm to activate certain proteins or phosphorylate certain protein to produce the desired action. But inositol triphosphate is soluble. It's not lipophilic, it's hydrophilic. Will be into the cytoplasm, okay, and will diffuse in the cytoplasm to bind to receptors of inositol triphosphate, which mainly present on the smooth endoplasmic reticulum to open calcium channels. So once inositol triphosphate bind to its specific receptors, especially on the smooth endoplasmic reticulum, it will increase the cytoplasmic concentration of the calcium and this will activate a cascade of kinases producing the intracellular activity or changes. Also, diacylglycerol and calcium work together to activate the PKC and phosphorylate the molecule to alter the cellular activity. يبقى تاني السيجنالينج باثواي ده اسمه phospholipase C signaling pathway اللي هو ده this is the effector activated by the G protein. Once the hormone bind to the G protein coupled receptor will activate phospholipase C, which convert phosphatidyl inositol biphosphate into diacylglycerol and phosphatidyl triphosphate. Diacylglycerol highly lipophilic, still within the membrane, and activate protein kinase C alone or with the calcium secreted by the effect of inositol triphosphate. Inositol triphosphate soluble in the cytoplasm bind to its receptors on the endoplasmic reticulum, increase the release of the calcium, increase intracellular calcium, which will activate also protein kinase C. This will produce the desirable effect by phosphorylation of the specific molecules, like proliferation, differentiation, apoptosis, cytoskeletal system remodeling, ion channel conductance, and neurotransmission. Calcium ions. Many cells respond to the extracellular stimuli by altering their intracellular calcium concentration. زي ما قلنا ممكن الكالسيوم لوحده or الفوسفاتيدايل inositol alone considered second messenger and can be the second messenger of many hormones alone, or both of them can be the second messenger. So calcium acts as second messenger by two ways: binding to the effector molecule like enzyme and activate it like protein kinase C, or bind to intermediary cytosolic calcium binding regulatory protein called calmodulin. Calcium will produce calcium calmodulin complex, which produce conformational changes in calmodulin and increase its affinity for the effector molecule. Once calmodulin be activated, it will cause contraction of smooth muscle. Calmodulin may activate also calcium calmodulin dependent protein kinases or may act directly on the effector protein. يبقى تاني الثيرد signaling pathway اسمه ايه؟ phosphatidyl inositol or calcium or both. اتكلمنا عن الفوسفاتيدايل inositol اللي هو بيجي من الفوسفولايبيز سي بيدينا diacylglycerol and phosphatidyl triinositol وقلنا both of them considered second messenger. الكالسيوم ions. Many hormones act by increasing the level of calcium ions inside the cell. Calcium is considered as second messenger by two ways. It activates certain effector proteins like protein kinase C with diacylglycerol to produce its desirable effect, or it will bind with certain calcium binding regulatory protein called calmodulin. Once calcium bounded with calmodulin, this will activate calmodulin, which will activate calmodulin dependent protein kinases or directly activate certain effector proteins. The effector proteins or target proteins of calmodulin including adenyl cyclase, phosphodiesterase, myosin light chain kinase, calmodulin dependent kinase, and calcineurin. اوكي، تعالوا نشوف بقى الرسمة اللي بتسهل لنا الفيجر. It will make it very easy to understand this signaling pathway. This is G-coupled, G-protein coupled receptor, and it's coupled with G-protein التايب بتاعه GQ, which will activate الإفكتور بتاعه اللي هو الفوسفوليباز C. Phospholipase C enzyme will convert الphosphatidyl inositol biphosphate into diacyl glycerol and inositol triphosphate. Diacyl glycerol maintained within the membrane and will activate protein kinase C, which will enter the cytoplasm and activate or phosphorylate more effector proteins. And الفوسفاتيدايل inositol triphosphate will be soluble into the cytoplasm, act on the receptor on the reticulum, endoplasmic reticulum, to open channels of the calcium. Calcium will be released intracellularly in the cytoplasm. Intracellular calcium will increase. Calcium will act with diacyl glycerol to stimulate protein kinase C, which stimulate or phosphorylate certain proteins to produce the desired action, or will complexed with calmodulin to produce calcium calmodulin complex. Once calcium calmodulin complex formed, this will produce conformational changes in calmodulin and activate it. Calmodulin will activate calmodulin dependent kinases, which phosphorylate also desired proteins اللي منها الميوسين والأكتين. This will produce muscular contraction and also or will affect directly the effector proteins to produce the desired action. يبقى ده الباثواي of phosphatidyl inositol or calcium or both. يعني ممكن ده يشتغل لوحده or الكالسيوم لوحده or both of them will work together. هنا الجي بروتين من النوع اللي هو جي كيو which stimulate phospholipase C. ناخد بالنا ان هو كده ده الشرح اللي من الاول بيجي لنا بعد كده في الفيجرز. This is a comparison or summarizing for the second messenger acting on G protein coupled receptors. كان عندنا الكالسيوم cyclic AMP, cyclic GMP, phosphatidyl inositol triphosphate, and diacyl glycerol. Calcium السورس بتاعه from the plasma membrane channels from the endoplasmic reticulum by the inositol triphosphate will increase intracellularly and act on calmodulin producing the complex اللي هو which activate calmodulin protein kinases and also will stimulate protein kinase C and will affect certain target proteins. مش لازم طبعا نعرف كل البروتينز دول، أنا بس جايباه to summarize ال second messenger السورس والأكشن بتاعهم. ال removal of the calcium by the pump or by the endoplasmic reticulum. Cyclic AMP السورس بتاعه الأدينايل cyclase bounded to the membrane from ATP will stimulate protein kinase A which produce its desired phosphorylation of a desired protein, and it will end by cyclic AMP phosphodiesterase. Cyclic GMP from guanyl cyclase له two types, membrane bound or soluble. It will activate protein kinase G and other proteins and end with hydrolysis by cyclic GMP phosphodiesterase. Inositol triphosphate comes from phospholipase C, which act on phosphatidyl inositol biphosphate and act on the receptor of inositol triphosphate on the endoplasmic reticulum, and its action ended by phosphatase. And diacylglycerol coming from phospholipase C also act on protein kinase C to produce its or activate phosphorylate its desired target proteins. طيب second signaling pathway group 2 hormones act on هو بروتين كاينيز كاسكيد. يبقى كده خلصنا الجي coupled بروتين receptors بال 3 second messenger بتاعها cyclic AMP, cyclic GMP and calcium or phosphatidyl inositol or both. في another signaling pathway اسمه بروتين كاينيزس. يعني ايه بروتين كاينيزس؟ احنا قلنا كاينيز يعني enzyme that add phosphate group from ATP to protein. ممكن this phosphate group will be added to tyrosine resist فيبقى اسمه tyrosine kinase, or added to serine and threonine فيبقى another type of kinases. تمام. طب يعني ايه cascade؟ protein kinase cascade is a series of protein kinase adding phosphate group to the next protein in the sequence. يبقى series بتعمل phosphorylation to each other, next to each other. The ligand bind receptor the same. This signaling pathway لازم الهرمون has to be bounded to the receptor, which lead to activation of الadaptor protein and lead to phosphorylation of a series of kinase. Protein kinase is very large family, more than 550 human type. And proteins are phosphorylated زي ما قلنا either on serine or threonine residues or residues of tyrosine, and this is called tyrosine kinase. Tyrosine kinase. Okay. يبقى from this explanation there is two types of the kinase cascaded. في tyrosine receptor kinases and non-tyrosine receptor kinases. First, a receptor tyrosine kinases, it has intrinsic tyrosine kinase activity. يعني الريسبتور itself has tyrosine residues and can autophosphorylate itself and phosphorylate other substrate and begin the cascade. Okay, it's part of the receptor protein. يعني الريسبتور itself has intrinsic tyrosine kinase. Example of receptors of that has receptor tyrosine kinases, receptor of insulin is the most important, and many other growth factors like epidermal growth factor, fibroblast, platelet derived growth factor, vascular endothelial growth factor, nerve growth factor, or non-receptor tyrosine kinase. So the receptor itself it's self-contained kinase, but it will recruit activity of intracellular protein kinases, which will begin the protein kinase cascade. يعني الريسبتور itself have no tyrosine kinase activity, but it can recruit the activity or can activate certain intracellular kinases to begin the protein kinase cascade. تعالوا نتكلم بالتفصيل أكتر عشان نفهم أكتر. الريسبتور تايروزين كاينيز قلنا ده those type of receptors اللي act on them الانسولين and many growth factor. التايروزين كاينيز action is on or off. Okay, once it's phosphorylated it will become activated and will begin activation of a series of protein kinase cascade to end at the target effector protein to produce the action. Tyrosine kinase receptors are family of receptors with similar structure. They have its own tyrosine kinase domain. يبقى have intrinsic tyrosine kinase activity تاني. يبقى tyrosine kinase receptors have intrinsic tyrosine kinase activity. It can phosphorylate itself. ده اسمه autophosphorylation, and also can phosphorylate other intracellular protein kinases to begin the protein kinase cascade. Structure of التيروزين receptors لها هرمون binding domain ولها carboxylic terminal segment. الcarboxylic terminal segment with multiple tyrosines for autophosphorylation وهنشوفها في الفيجر حالا. When hormones bind to the extracellular domain, the receptor will aggregate and will dimerize. Dimerize يعني ايه؟ dimer يعني ايه؟ يعني two. يعني once الهرمون bounded to the tyrosine receptors, it will aggregate and dimerize, then will be autophosphorylated in a tyrosine residues in the carboxylic terminal end of the receptor. Autophosphorylated tyrosines will has high affinity binding site for many intracellular signaling protein kinases which contain حاجة اسمها ايه SH2 domain اللي هي الساركوما homology domain. SH2 domain ده one of the abbreviation برضو اللي هنستخدمها النهاردة. تعالوا نشوف على الفيجر هيفهمنا اكتر. هنا ده اسمه التيروزين ريسبتورز تيروزين ريسبتور كاينيزز لها extra cellular domain which bind to the ligand اللي منها الهرمونز منها الانسولين ولها carboxylic and intracellularly فيها التيروزين residues. It has its intrinsic tyrosine kinase activity so it can autophosphorylate itself. Once الهرمون bind to the receptor it will aggregate and dimerize يبقوا اتنين كده مع بعض. بعد كده بيحصل autophosphorylation of التيروزين residues موجودة in the intracellular part of the tyrosine receptor. Then it will have high affinity to many intracellular kinases to begin the protein kinase cascade. It will activate many protein kinases like the GRIP, the SOS, RAS, RAF, MIC, ERK. All of them are kinases, and the MAP kinases cascade, it's very important kinase, and the ERK and many transcriptional factor to produce its desired action like proliferation, differentiation, survival, and cell migration. we will give an example for receptor tyrosine kinase. The most important and famous example is the insulin receptor tyrosine kinases. Insulin bind to the receptor will cause conformational shift, and this will promote triple autophosphorylation of the cytoplasmic beta subunit of the tyrosine kinase receptor. Autophosphorylation will trigger movement of activation loop, which expose the substrate binding site of the B subunit of the receptor. Once activated, the insulin receptors will bind and phosphorylate the insulin receptor substrate IRS1, which then serve as the core of multi-protein complex. IRS1 will bind to the GRIP2, and the GRIP2 have a structural domain SH2 domain. Then GRIP2 bind to the SOS via its SH domain, and SOS bind to the RAS protein, which cause it to bind to GTP and release GDP. كل دي الكاسكيد اوف بروتين كاينيزز، كل دول كاينيزز. تمام، نقولها تاني ان الانسولين bind to a receptor، يحصل dimerization، autophosphorylation، exposure of the substrate binding site in the intracellular part of the receptor will activate أول حاجة إيه؟ insulin receptor substrate 1، then GRIP2، then SOS، then RAS. RAS protein have GT-base activity. GT-base activity جات لنا قبل كده في الجي بروتين. يعني ايه GT-base activity؟ it have the ability to exchange between GDP and GTP nucleotide. Okay, once it's active it will be connected to GTP nucleotide, then it will activate RAF1, and RAS will activate phosphorylation of a cascade, another cascades like MAP kinase, ERK. Activation of certain kinases result in both transient immediate response or prolonged delayed response. قلنا RAF 1 will phosphorylate MEK and MAP kinase, and ERK, which enter the nucleus and phosphorylate transcriptional factor to affect cell division. يبقى binding of the insulin to the alpha subunit اللي هي extracellular subunit of the insulin receptor will produce autophosphorylation of the beta subunit of the insulin. This autophosphorylation will expose the substrate binding sites in the insulin receptors and will phosphorylate insulin receptor insulin response substrate 1, then the cascade GRIP2, SOS, RAS, RAF, MAP kinase, ERK, and MEK. All will produce the physiological action of the insulin, which will affect gene expression, metabolism, and growth. تعالوا نشوف الرسمة. First, insulin receptor bind to the insulin will bind to the insulin receptor. بيحصل بعد كده autophosphorylation, triple autophosphorylation in each receptor. Then the insulin receptor will phosphorylate insulin response substrate 1, which will begin the cascade GRIP2, SOS, RAS, RAF, MEK, ERK, and also MAP kinases. All of them contain SH2 domain. Those proteins contain SH2 domain and will be activated and phosphorylated to produce at the end the ERK enter the nucleus to control transcriptional factor and produce or control cell division. هنا insulin alpha subunit and beta. Okay. This is tyrosine kinase receptors have the ability of autophosphorylation, triple autophosphorylation, phosphorylate the iris, then begin the cascade any protein which contain SH domain like GRIP2, SOS, RAF, MYC, and MAP kinases. MAP kinases will produce activate transcriptional factor which are anti-apoptotic and control differentiation and also it will control many metabolic pathway like glycogen synthesis, protein synthesis, glucose uptake, anti-lipolysis, many physiological action of insulin. The last part of our lecture is non-receptor tyrosine kinases. احنا قلنا عنها ان هي it has no intrinsic tyrosine kinase activity but it has the ability to trigger or recruit intracellular kinases and activates them. والإكزامبل عندنا الجاك stat receptor pathway which is stimulated by growth hormone, prolactin, erythrobiotin, interferons, interleukins, and many cytokines. Receptors have no intrinsic activity, so it can't autophosphorylate itself, but it will trigger or recruit intracellular many intracellular tyrosine kinases like الجاك kinase اللي هي genus kinases. Receptors will mediate dimerization also, and then phosphorylation of the STAT اللي هي signal transducer and activator of transcription. Then STAT will dimerize and translocate to the nucleus and regulate transcription. There is about 4 JAKs and 6 STATs which combine differently for different cell types and signals. Upon ligand binding, these receptors dimerize and interact with cytosolic tyrosine kinase JAK, which will autophosphorylate and phosphorylate other tyrosine kinases. It will phosphorylate. It will phosphorylate the STAT protein signal transducer and activator of transcription, which will traverse the nuclear membrane, dimerize and translocate to the nucleus, bind to specific DNA element to regulate transcription. Also, it can phosphorylate MAP kinase cascade also. تعالوا نشوف الرسمة. هنا those receptor itself, it have no intrinsic tyrosine kinase activity. It can't autophosphorylate itself, but it can trigger certain tyrosine kinases اسمها ايه؟ الجاك. Okay, and SOX. الجاكس once الهرمون bind to the receptor, it will dimerize and phosphorylate the JAK, which will phosphorylate STAT, and then STAT will dimerize, phosphorylated STATs will dimerize and traverse nuclear membrane to bind to certain binding sites on a DNA and control transcription of the desired genes. Here also this is hormone اللي هو مثلا for example growth hormone or prolactin. This is non-receptor tyrosine kinase. It can't autophosphorylate itself, but it can phosphorylate or trigger phosphorylation of certain intracellular kinases. أول واحد فيهم اللي هو الجاك will phosphorylate, and then JAK will phosphorylate the STAT. STAT will dimerize once phosphorylated, enter the nucleus, bind to DNA to control transcription. And this is the last signaling pathway by hormones with its examples like prolactin, growth hormone, cytokines, interleukins, and I hope you understood, and I'm waiting for any modifications or any questions from you, and thanks.